I will pass the word to Valentina LIBRI, who will discuss single-cell transcriptomics. Valentina?
Hi, I'm just going to share my presentation. Can you see my my presentation on?
Yes.
Yes.
Okay. So, as you saw, Milena gave a nice overview of the technologies that are that have a single cell resolution. I will focus my talk on technologies that are whole genome and next generation sequencing based. So whatever is the interest or the omic that you're going to explore, there's one thing that is common to all the approaches, and is the sample preparation. So this is a key step that is often underestimated, and having a good single cell preparation is key to get good quality data. So in the sample preparation, there's the first step of dissociation that can be easier in samples like blood, that it can be just isolated as single cell suspension with a gradient, or more complex complex in tissues that have to be dissociated either by mechanical or enzymatic procedure. So you have to find the best procedure for the tissue of interest and ensure that your cells are in a good health. So you have to consider also if after obtaining your single cell suspension, if you need an enrichment step of your cells or not, depending if you're looking at a rare population, and you need to enrich by FACS or magnetic separation. Or if you're, for example, you have a larger percentage of population, or you're interested in looking at the heterogeneity of your sample. Of course, you have to perform pilot study to define the best solution, and also the number of cells that you have to deal with. And there is a nice link on Satya's site that supports you in taking a decision there. So after that, of course, you choose your technology, your preferred technology. We're going to go through all those during my talk. And what one has to keep in mind all the time is that sample processing might and often induce bias in your data, and then your manipulation has to be as rapid as possible. Often the work on single cell is done on fresh samples, but there's also the need to develop system of preservation. And this is because, for example, if you want to analyze a time course or different donors, and you do want to have batch effect, and you want to analyze them at the same time, or if you do not have your equipment in your institute and your sample has to travel, or if you're dealing with samples from clinics that can come at any time of the day, or if you're having like samples that are stored and you want to analyze later on. So there are several ways that have been tested to preserve the samples. A lot is used cryopreservation and freezing. For freezing of entire tissues, it's have no problem, for example, for a single nuclei RNA-seq. For other fixative, or for fixative, not all are adapted to all kind of cells, and you have to consider that you can always introduce a bias in your data, and this has to be considered and maybe compared, if possible, to fresh data. Fresh samples, sorry. So the omic that is mostly developed, as Milena was saying, is the transcriptomic. And there have been in the last year an explosion in the development of single-cell techniques, RNA-seq techniques, and also an exponential number in increase in the number of cells that can be analyzed in one experiment, as you see here. I won't consider that one has to necessarily use what has the highest throughput, and that has been developed most recently, because there are advantages and disadvantages on high throughput, and there's a trade-off with sensitivity. So which one of the technology you are going to use, and on which parameter do you base your choice? So to do that, I'm going to go through different technologies. All of them have a same kind of pipeline, same steps as you see here in the slide. First is the cell isolation. So you have to partition at the beginning your cell, and it can be either through FACS, and that is partitioning the cell in a well of 96 well plate or 384. This is in general the low throughput systems. There's an integrated fluidic circuits that it was developed by Fluidigm in 2013. Still a lower throughput, and then there are droplet system in which the partition is the droplet or microwell, and of course is the wells, or the split pool in which the partition is interestingly the cell itself. So for the second strand synthesis, the main system used is the template-switch oligo. This system is based on the ability of the retrotranscriptase to add non-templated cytidine after retrotranscribing. And in the presence of the template-switch oligo that carries the guanine and is annealing to the newly synthesized DNA, the retrotranscriptase is able to continue and switch to the DNA to copy. And so you will have a cDNA newly synthesized with a PCR handle that will be common to all the transcripts. An alternative to this is the polyadenylation. So there is a poly A tail added to the end. So your PCR oligo will carry a poly T stretch. Of course, you need a cDNA amplification, as the initial material from a single cell is really limited. You will be able to amplify your material because you added a common handle to the extreme of all the cDNAs. And you can do it either by PCR, that allows you to have a big amplification, but you might introduce bias. Or like you can introduce less bias with a linear amplification, so the in vitro translation, but it's a slower process. Of course, you have a step of fragmentation. So you have cDNA that are long kilobases, but the common sequencers that have a right throughput for RNA sequencing can analyze only a few hundred bases. So you need to fragment your cDNA, and this can be done either by fragmentation or fragmentation by enzymatic or mechanical fragmentation. So what you will obtain are libraries that you will sequence, and those will be either full length, so you are sequencing the whole length of the cDNA, or only sequencing the 3' end or 5' end. The full length is typical of the low throughput technologies. So if we go through those approaches, in my presentation, you will see I always take as a reference one of the technologies that is mostly used with the reference, but I keep on the bottom of the slide references for similar technologies if you're interested in. So the SmartSeq technology, improved in SmartSeq 2, is use FACS to sort cells in plates. And what you notice is that the cells are handled one by one, meaning that from the beginning to the end, the cells are separated. And this means that you have a very high sensitivity and coverage in this technology, that you can detect the splice isoforms and the SNPs, and even in immune cells, the full-length B and T cell receptor. But on the other hand, it's very labor intensive, even though can be automated. There is a high cost per cell, and it is still low throughput. And there's no unique molecular identifier. So the UMIs are sequence that are degenerated and are present in the barcodes, and those sequences can account for PCR bias. So this cannot be used in the full-length approaches. So an intermediate throughput we have with the tag-based approach with, for example, MARSEC, that was developed at the Weizmann Institute. Here you see that you can have early at the level of the retrotranscription the barcoding of the transcript. So you can after that pull the wells together, so reducing the handling. This reduces the cost. An important thing, you can link the transcriptional profile to the FACS profile of the cell. This is true for the SmartSeq. For similar technologies, and it can be automated, but still it's a medium throughput. And the disadvantage is that you reduce the sensitivity, and it's time consuming because the library preparation can take up to 1 week. An interesting tag-based approach is the split-seq that has been developed in 2018. As I said, here the cell acts as a compartment. So the cell is fixed and permeabilized, and you have a retrotranscription in situ. After that, cells are split in different plates, and each plate there is an in-situ ligation with a specific barcode. After that, cells are pulled together, split, and again a second ligation step. And this is reiterated so that you have a different sequence combination of barcodes that gives unicity to the cell. And this gives a potential high scalability. You can analyze many samples at the time, and you do not require any specific instrumentation. And this is compatible with fixed cells. Of course, this is on the other side, it's a disadvantage. As we said, fixation can introduce a bias. There's a high cell loss, so the initial one has to take in account this as in the initial amount of material that one has to start with. And there can be some background at the level of the analysis that might be improved in the last year. Another step that was quite important in the development of single-cell approaches, because improved the throughput, is the droplet approach, in which Cells are mixed with beads that are carrying a specific and unique barcode on the surface in aqueous solution, and this mixed with oil, creating small droplets. So the cell is captured in one droplet with a specific barcode, and so we can at this point lyse the cell, and all the mRNA coming from this cell will be distinguished by the specific barcode. And so here we can then pull them all together and process all together. So as the barcoding is happening very early, we can have just one library preparation per sample. And this becomes very high throughput because we can process a high number of cells in parallel. Of course, we have UMIs, and this procedure lowered the cost per cell. And there are many or several commercial solutions that are easy to use with this system. On the other side, there's we reduce the sensitivities as we increase so much the number of cells analyzed, and we need also a specific equipment for that. An important point that I want to make is also that we cannot take imaging on these cells, and this means that we cannot analyze doublets. We have to assume the percentage of doublets, but we cannot visualize them, neither exclude them. And the commercial solutions that are still have a high cost per sample. So there are several commercial solutions for RNA-seq that have been developed and that are available on the market. The C1 was one of the first in 2013, based on this integrated fluidic circuits that allowed an on-chip process until the cDNA. But this had a low throughput and also low capture rate. So now it's not very much used, even though the sensitivity was quite good. The Chromium and the Nadia instead are based on droplet, and they have high number of cells that can be selected, and there's a difference in the size of the maximum size of the cells that can be selected there. There are also other two possibilities of two machines like the Isolate and Chelsea, based on nanowell and separation by gravity, and those allows the visualization and imaging of the cells, getting also morphology and selection of the cells. So I would like to highlight briefly the 10x because it was quite successful in the last year. There are many works that have been done on the 10x that came out in 2016, and it was quite productive in integrating, implementing new applications like gene profiling 3' end, the immune profiling at 5' end, the feature barcoding that allows also the concomitant detection of surface protein, detection of copy number variation, and epigenomic study with UTechS. And really recently, end of 2019, it came out with a spatial transcriptomic that we will discuss later on. A nice application that I want to highlight with the next was this combination of the RNA-seq, single-cell RNA-seq, with the CRISPR technology. So in the same cell, it was possible to identify a specific perturbation of one gene and correlate directly with the modification in the transcriptional profiling of that cells, and this allows also so the selection of a pool of perturbers. So at the end, looking at all the transcriptional single-cell RNA-seq approach, what we have to consider when we choose our technology that suits our scientific question, we have to consider the sensitivity. As I told you, if you're looking at very lowly expressed genes, you need to go for sensitivity. If you need to look for very rare cells, of course, you go for high throughput, and you have to see if it's relevant for you to have information on isoforms or SNPs. If you are considering, for example, that it's very important for you to have singlets, then, for example, you cannot use technologies that don't allow you to visualize and select the cells that are captured. Of course, the cost of the technology has always to be considered with that. So, as Milena was saying, the next step in the single cell technologies is to try to put together information, integrate information from different omics to get to a different level of investigation. And so the transcriptomic has been linked with other applications, like with the protein detection. There are several technologies that have been developed. Some have very low throughput also for the RNA, so they are not whole genome for the transcriptomic. So I will concentrate only on the whole genome for the transcriptomic part. So one of those is based on the use, so the detection of surface protein, and is based on the use of antibodies that are coupled with a barcode. This has been used, for example, in the CytoSeq technologies. This allows to stain the cells with the antibody and select with a normal droplet system experiment. But when you capture the cell, together with the cells, you have the antibody, and this will be prepared as a library together with the transcriptomic. So, and it will carry the same barcode of the cell. So you will have a library for the transcriptomic, a library for the antibody, and they will carry the same barcode. So this can link to the same cell and put together the information. The cell hashing was developed in the year after, and it's exactly the same principle, but here we have the same kind of antibodies, just different oligos that are coupled to it, and this allows to treat different samples with those antibodies and pull them together. This allows either to reduce costs, or also it allows you to identify, to reduce bias, batch effect, and identify doublets. The RNA-seq, it's a nice example on how you can get the maximum of information in a similar experiment. In this one, for example, it was possible to get information of the transcriptomic and TCR-BCR, so the T-cell receptor, B-cell receptor specificity or expression, and the cell surface protein together with the CRISPR perturbation. So the cell has a function, but it depends on its localization and the environment in which it is. And doing this kind of experiment, we lose the information, we lose this connection with the environment. So the need is to understand the cell the function of the cells inside their the original location. And this is what is addressed by the new technologies as spatial transcriptomics. So spatial transcriptomics was a technology developed in 2016, and now acquired. The company was acquired by the next Genomics. In this system, you have a slide on which you have spotted oligos that are carrying barcodes and that capture the transcript. So as you put the slice of your sample over the slide, you can permeabilize your sample. The RNA will be released and captured by the oligos spotted on the side. Of course, each oligo will carry a barcode that will be carrying the information of the specific position on the slide. So on the slide, you carry on cDNA synthesis and library preparation, so that after analyzing the data, not only you have the sequencing and the gene expressed, but you also have the information on the position in which they were originally in the tissue. So the resolution of this technology is not yet single cell, so for the moment is about 55 micrometers, so few cells depending on the size. And there's a new development in 2019 of the slide-seq technology, in which the oligos are spotted on beads of 10 micron, and possibly the size can be reduced, so the resolution is higher. The process is kind of similar to the one for spatial transcriptomic in principle. And there's a new development, the high sensitivity spatial transcriptomic, in which the resolution went down to 2 micrometer. So one thing with which I would like to finish is the validation. So these are all high throughput technologies with a certain level of sensitivity. And it's really always important to validate these results with the biomarker that have been identified after bioinformatic analysis, and validate with technologies that have in which you validate a reduced number of biomarker, but with a higher sensitivity. With this, I would like to thank you, and I would take any question.